Hello everyone, welcome to the new session of Genetics and Plant Breeding, Cell and its Structure. This is the third part. Okay, so in this session, we are going to cover up the endomembrane system. Endomembrane system. So what is the endomembrane system? We are going to study in this session, right? So each of the membranous organelles, the organelles which contain membrane, right? Membrane bound organelle is distinct in the terms of its structure and function. So very, very important. All the organelles, all the membrane bound organelles, right? Some membrane bound organelles are spherical in structure, some are in the layers of stacked structure, some are forming the network like structure, right? So there are different types of membrane bound organelles. So they are different in the terms of their structure and their function. Many of these are considered together as an endomembrane system because their functions are connected, but there are some organelles that unitedly they do a particular function, right? So that is why they are considered as a endomembrane system. So the endomembrane system include endoplasmic reticulum, Golgi complex, lysosomes and vacuoles. So we are having 4 different organelles. They are endoplasmic reticulum, Golgi complex, lysosome and vacuoles, and they form endomembrane system, right? So how they are unitedly interconnected to perform a particular function, we are going to see that. Since the function of the mitochondria, chloroplast and peroxisomes are not coordinated with the above compartment, these are not considered as a part of the endomembrane system. So there are some other organelles also. They are also membrane bound organelles like mitochondria, chloroplast, peroxisomes, okay? But they are not the part of the endomembrane system. They are not the part of the endomembrane system because they are not in a coordination to perform the function that endoplasmic reticulum, Golgi complex, lysosome and vacuoles are performing, right? So they are nothing to do with this system. Electron microscopic studies of the eukaryotic cell reveal that presence of a network of a reticulum of a tiny tubular structures scattered in the cytoplasm that is called as endoplasmic reticulum. So the first organelles of the endomembrane system is the endoplasmic reticulum. So how is the endoplasmic reticulum? Endoplasmic reticulum form a network, tubular network like structure like this. So these are the tubular structure, right? And they are also connected with the nuclear membrane. So this is the nuclear membrane. And forming the nucleus. Like this. So this is the nuclear membrane. This is the nuclear pore structure, and this is the endoplasmic reticulum. Tubular structure and interconnected, right? They are present in the cytoplasm. ER divides the intercellular space into 2 distinct compartments. Okay, so the luminal inside the ER and the extraluminal site that is present in the cytoplasm compartment. So we see that this particular lumen of, this is the lumen of ER. So by forming this endoplasmic reticulum, there is a compartment which is present inside the endoplasmic reticulum that is called as luminal compartment, or that is present inside the ER, and the one compartment that is present outside the ER, that is the cytoplasmic compartment. Okay, the ER often shows ribosomes attached to their outer surface. So sometimes the endoplasmic reticulum surface is attached with the ribosomes. They are attached with the small ribosomes. Okay, so they gives a rough appearance at their surface. They gives the rough appearance at the surface. So that is called as RER, means rough endoplasmic reticulum. The endoplasmic reticulum bearing the ribosomes on their surface is called rough endoplasmic reticulum. Okay, so RER frequently observed in the cells, frequently observed in the cells that are actively involved in the protein synthesis and secretion. So they are mostly present in the cells that are actively involved in the synthesis of protein and its secretion. So here you can see the structure of RER. This is RER, rough endoplasmic reticulum. All these small ribosomes are attached to the surface of the endoplasmic reticulum. They are extensive and continuous with the outer membrane of the nucleus. So here you can see they are attached to the outer surface of the nuclear membrane. Okay, that is the nucleus, and they form a tubular network all over in the cytoplasm. And the ribosomes are attached to its surface, forming the rough endoplasmic reticulum. Okay. The smooth endoplasmic reticulum is the major site for the synthesis of lipid. Okay, so the endoplasmic reticulum that are not having any ribosome attached to its surface is called as the smooth endoplasmic reticulum, and that is considered as SER. SER means smooth endoplasmic reticulum. They are the major site for the synthesis of lipid. In the absence of ribosome, they appear smooth and are called as smooth endoplasmic reticulum. In animal cells, lipid-like steroidal hormones are synthesized in the SER. So, they are mainly responsible for the formation of the steroidal hormones in the case of animal cells. Now, the second organelle of the endomembrane system is the Golgi apparatus. Golgi apparatus. Camillo Golgi, 1898, first observed densely stained reticular structure near the nucleus. So they are present near the nucleus. So suppose this is the nucleus. Golgi apparatus is present like this, near the nucleus only. Golgi apparatus. Like this. Okay, so they were later named as Golgi bodies after him, after the name of the discoverer. This organelle got its name Golgi bodies. They consist of many flat disc-shaped sacs or cisternae. So here you can see a disc-shaped flat cisternae. Okay, they are the sac-like structure, disc-shaped, flattened, sac-like structure which are called as cisternae, and they are of 0.5 micrometer to 1 micrometer in diameter. So they are stacked parallel to each other. So they are the sac-like structure, that is the cisternae. They are arranged parallel to each other, and varied number of cisternae are present in the Golgi complex. So there are not there, the cisternae number is not fixed. So varied number of cisternae are present in the Golgi complex. The Golgi cisternae are arranged near the nucleus with distinct convex cis or the forming endoplasmic reticulum, cisternae phase, and the concave trans or the maturing phase. So here we can see that if this is the cis phase, so this will be the trans phase. Now what happened? Suppose this is the endoplasmic reticulum, right? It form a protein-like structure which is present in the vesicles. So this vesicle comes and fuses with the cisternae of the cis phase. And the protein which is present in this vesicle now get enter into the cisternae, right? So there will be the modification of this particular protein in this cisternae, and this modification occur at the different levels, means it will again pinched off and sent to the next level of cisternae. Certain modification occur here also, and again it is pinched off and sent to the third level. Like this, it is sent to the last level of the cisternae, and from there, the trans phase, it reaches to the trans phase of the cisternae, and it pinched out a vesicle. Which is having mature protein, So cis phase and trans phase are entirely different. Their structure is different, their composition is different, right? The Golgi apparatus principally performs the function of packaging material. So what is the main function of the Golgi apparatus? The main function of the Golgi apparatus is the packaging of materials that has to be delivered either in the intracellular targets or secreted outside the cell. Okay, so the main function of the Golgi apparatus is to packing of the material that is given out from the endoplasmic reticulum. Material to be packaged in the form of vesicles from the ER fuses with the cis phase of the Golgi apparatus and move toward the maturing phase. This we have already discussed. This explains why the Golgi apparatus remains in close association with the endoplasmic reticulum. So now we understand that why this Golgi apparatus is a part of endomembrane system. They are highly connected to the endoplasmic reticulum, which is also a part of the endomembrane system. So the thing which got manufactured or synthesized, that is the protein in the endoplasmic reticulum, is transferred to the Golgi apparatus for its packing or modification, right? So a number of proteins synthesized by the ribosome on the surface of the endoplasmic reticulum are modified in the cisternae of the Golgi apparatus before they are released from its trans phase. So endoplasmic reticulum and Golgi apparatus are highly interconnected. Golgi apparatus is the important site. It is the important site of formation of glycoproteins and glycolipids. So glycolipids are synthesized in the SER, whereas the glycoproteins are synthesized in the RER, right? But their modification, their formation or attachment of the carbohydrate to the protein moiety of any structural protein occurs in Golgi apparatus. So protein is synthesized in the RER, but attachment of this glycoprotein, attachment of this carbohydrate part to the glycoprotein occur in the Golgi apparatus. In the same way, lipids are synthesized in the SER, but this carbohydrate part of the glycolipid get attached in the Golgi apparatus. So these are the membrane-bound vesicular structures formed by the process of packaging in the Golgi apparatus. Okay, the third one, lysosome. Lysosome, very important. The isolated lysosomal vesicles have been found to be very rich in almost all types of hydrolytic enzymes. So this organelle is very, very rich in the hydrolytic enzyme. For examples, hydrolases, okay, like lipases, proteases, carbohydrases. So these enzymes are present in the lysosome and optimally active at the acidic pH. So they're required for their activity a certain pH, and generally it is acidic pH. So these enzymes are capable of digesting carbohydrates, proteins, lipids, and nucleic acid. So how they are important? Now you can understand that they are responsible for the digestion of carbohydrates, digestion of proteins, lipids, as well as the nucleic acid. The vacuoles, the fourth one, that is the vacuoles. The vacuole is the membrane-bound space found in the cytoplasm. Okay, it contains water, sap, and excretory products and other materials not useful for the cell. So whatever the substance gets synthesized as a byproduct, not as a main product, but as a byproduct, which is not important or which is not useful for the cell, that gets stored inside the vacuoles, So whatever the excretory products, sap and water get synthesized, extra water, okay, that get stored in the vacuoles. The vacuole is bound by a single membrane called tonoplast. So this is a very important. The membrane of the vacuole got a specific name that is called as tonoplast. The plant cell, the vacuoles can occupy up to 90% of the volume of the cell. Very, very important. So 90% of the volume of the plant cell is occupied by the vacuole. Okay. So in plants, the tonoplast facilitate the transport of the number of ions and other materials against the concentration gradient into the vacuole. Hence, their concentration is significantly higher than the vacuoles than in the cytoplasm. So all the extra material which is not required by the cell gets stored inside the vacuole, and that too against the concentration gradient, right? In amoeba, the contractile vacuole is important for the osmoregulation and excretion. So whatever the extra water gets stored inside the cytoplasm of the amoeba, it is transferred to the contractile vacuole, and this contractile vacuole pinched off the water from this contractile vacuole out of the cell. So it is helping in some sort of excretion, right? So whatever the function in human the kidney is doing, the contractile vacuole is doing the same thing in amoeba. In many cells, as in protist, food vacuoles are formed by engulfing the food particles, right? So as we see in the case of amoeba, when food comes in contact to the amoeba, so this is amoeba and this is the false food, that is pseudopodia. So food is present here. Now this pseudopodia engulfs the food, forming food vacuole. This is the nucleus, right? So whatever the enzymes are present in the cytoplasm is given to this food vacuole and food get digested. Okay, now after the digestion of food, whatever the digested food is there, it get absorbed to the cytoplasm, and whatever the undigested food present, it given out from the cell out, that is called excretion, right? So in this way, food vacuoles are formed in the case of amoeba. Okay, so this is all about this session. We have completed the endomembrane system. It means that we have studied about endoplasmic reticulum, Golgi apparatus, lysosome and vacuoles. Okay, some of the organelles we are going to study in the next session. Till then, thank you very much.